As introduction, agarose gel electrophoresis is one of the laboratory methods that we will use to separate mixtures of DNA, RNA or proteins according to the molecular size. Principle: the molecule to be separated are pushed by an electrical field through a gel that contains a small pores. The molecules travel through the pores in the gel at a speed that is inversely related to their length. This means that a small DNA molecule will travel a greater distance through the gel than will a larger DNA molecule. The movement of charged molecule is called as a term migration. Molecule migrate toward the opposite charge. A molecule with a negative charge will therefore be pulled toward the positive end. In fact, DNA and RNA are negatively charged molecules, so they will be pulled toward the positively charged end of the gel.
Polyacrylamide gel electrophoresis is a technique used to separate a mixed sample of proteins to identify and quantify single proteins from the mixture. The most widely used type of electrophoresis on polyacrylamide gel is sodium dodecyl sulfate polyacrylamide electrophoresis, or also known as SDS-PAGE, which is used to separate proteins. It is also possible to use PAGE to separate DNA and RNA, but proteins are the most common sample type. The principle is based on the concept that charged molecules move through an electric field to an electrode that has a sign opposite to it. The detergent sodium dodecyl sulfate is used to denature the proteins and normalize their mass to charge ratio. The proteins that are coated by SDS are charged negative, and when loaded onto a gel and put within an electric field, they will move toward the electrode that is positively charged. It is then separated by a molecular sieving effect that is based on size. After visualization using staining, which is protein-specific staining method, the size of the protein could be estimated by comparing the movement distance to that of the molecular weight data.
The advantage of the agarose gel electrophoresis. First, the agarose gel does not denature the DNA sample and they stay in their own form. The main benefit of the agarose gel technique is that it can be easily processed and the DNA molecule that is used as a sample can also be recovered without any harm to it at the end of process. Next, agarose gel are simple and rapid to prepare. For most applications, only single component agarose is needed and no polymerization catalysts are required. Then, the gel is easily to pour and the pore size of the gel is large enough to run DNA or RNA. Disadvantage of agarose gel electrophoresis, such as gel can make during electrophoresis with current is passed through it, lead the chance that genetic material can adopt the shape which are not needed. Then different form of genetic material may run in unpredictable form due to genetic variation or mutation in DNA. Next, the buffer used can be exhausted. For example, TAE, triacetate EDTA buffer has a lower buffering capacity which can lead to exhaustion by repeated use. Then, the advantage of PAGE, such as the pore of the polyacrylamide gel, can be altered in easy and controllable fashion by changing the concentration of two monomers. Next, greater revolving power. It also can accumulate largest quantity of DNA without significant loss in resolution. Then, stable chemically crystalline gel and good for separation of low molecular weight fragments. Lastly, the DNA recovered from polyacrylamide gel is extremely pure. Then, the disadvantage of PAGE, such as toxic monomer which are acrylamide gel, is neurotoxic in nature. Next, gel are difficult to prepare and often leak. It takes a longer time for preparation and handle more than agarose gel. Next, pore band resolution due to high alkaline operating pH.
There are a few similarities between agarose and polyacrylamide gel electrophoresis. The first one is both electrophoresis is a laboratory technique that separates molecules based on their charge and size. Second is both electrophoresis use electrical charge in order to enable the molecules such as DNA move through the gel. Molecules with a negative charge will be attracted to the positively charged anode, while molecules with a positive charge will be attracted to the negatively charged cathode. Third, both of these methods need buffers to provide ions that carry a current and to maintain the pH at a relatively constant value. These buffers have a plenty of ions in them, which is necessary for the passage of electricity through them. Next, this method is also used the same casting apparatus that consists of tray, the support and combs. The tray is the actual mold which provides a shape for the gel as it polymerizes. Lastly, both techniques use a gel step with small pores through which macromolecules move.
Firstly, based on the origin, agarose is a natural polymer made of marine-based polysaccharides extracted from the agarosides of rhodophyceae marine algae. Meanwhile, polyacrylamide is a synthetic polymer created by free radical polymerization of acrylamide monomers with bis-acrylamide producing polyacrylamide. Molecular complexity. Agarose is a complex polysaccharide that exists as a linear polymer with repeating units of agarobiols. In contrast, polyacrylamide has a simple repeating linear chain structure with acrylamide subunits, but the linear structure converts into a cross-linked polymer through the bis-acrylamide, creating cross-link between the acrylamide monomers. Chemical bonds. Hydrogen bonds hold agarose molecules together, making the agarose gel less stable. Meanwhile, the acrylamide molecules of polyacrylamide gels are held together by covalent cross-linking, making them chemically more stable than agarose gel. The pore size of the gel. Agarose has a somewhat larger and a non-uniform pore size. Its pore size becomes smaller with the increasing concentration. Pore size of agarose gels can be regulated by increasing or decreasing molecular sieving. In contrast, polyacrylamide gels have a small and uniform pore size, making them more suitable for separating proteins over large polynucleotide DNA or RNA fragments and allowing the separation of relatively small proteins. This gel's pore size can also be altered more controlled manner than agarose gel. The ratio of acrylamide to bis-acrylamide determines the pore size of the gel. Gel preparation, namely the pouring orientation. Agarose gel is poured horizontally, making it easier, and agarose also can be heated to aid the pouring process, but it should not be heated to such an extensive degree that the agarose ends up melting. On the other hand, polyacrylamide is poured vertically with the help of a glass plate as an additional support because its structure is very thin, and unlike agarose, polyacrylamide cannot be reheated and then poured.
The steps involved in agarose gel electrophoresis and batch are more or less the same. The difference lies in how the gel is prepared and how it is casted. In agarose gel electrophoresis, the first step is to prepare the tris acetate EDTA buffer, which will help maintain the pH of the sample. The buffer is then heated and 2 grams of agarose powder is mixed into the solution, and the resulting solution is then transferred into a water bath to cool down, and once it has cooled, ethidium bromide is added into the solution, and then the solution is poured into a mold and a comb is inserted to create wells. Once the gel has completely set, the comb is removed and the gel is submerged into 1 mm depth of electrophoresis buffer, and the same buffer is used and mixed with the sample, and the sample is poured slowly into each well using a pipette. The electrical leads are applied to both ends of the chamber and the voltage is run. Once the sample has migrated and the electrophoresis process is done, the gel is removed from the chamber and stained to be viewed under the UV trans illuminator. As for batch, the sample is first mixed with a denaturant such as SDS or urea depending on the sample used. The sample is then heated to up to 95 degrees Celsius in order to enhance the denaturation process. Then a tracking dye is added into the sample in order to mediate tracking of the electrophoresis progress. Then the separating gel and the stacking gel are prepared by mixing distilled water, acrylamide, tris, SDS, ammonium persulfate, and phenol. The first and the main layer is the separating gel, which is poured in between the glass plates, and this layer is sealed with water in order to prevent contact with air that will inhibit polymerization. Once the gel has completely polymerized, the water is poured out and the excess water is taken out by absorbing with a filter paper. Second and the shorter layer is the stacking gel. This gel is poured on top of the separating gel and a comb is inserted. The comb is removed when the gel has set and the sample is loaded into each of the well. the voltage is applied, and once the electrophoresis process is done, the gel is removed from the glass plates and stained for observation.
DNA ladder is a molecular weight size marker, also referred to as a protein ladder or RNA ladder, which is a set of standards that are used to identify the approximate size of a molecule run on a gel during electrophoresis. In order for us to further confirm the location of where we load the samples, simply by looking at the DNA ladder. The largest fragment will be located on the same side as the sample load, where it will go in the direction of the smaller fragments. Hence, we can see from the figure that we load the sample from above, as the largest fragment of the 1500 base pair is located on the upper side and is moving in a downward direction. Using the principle that molecular weight is inversely proportional to the migration rate through a gel, where the heaviest fragment will travel less distance as compared to the lighter fragments. The first result interpretation is based on the restriction digestion. The figure exhibits an example of DNA restriction digestion with Nia3 endonuclease, which will further cleave mutant DNA that contains a restriction site. Firstly, lane 6 is known to be the molecular marker. We can observe that lane 1 and 5 are categorized as homozygous normal, as the normal allele remains uncut due to the absence of a restriction site for Nia3 endonuclease, hence exhibiting a single tip band of 400 base pairs. Next, lane 2 and 4 are categorized as homozygous mutant, as mutant allele contains restriction site for Nia3, which further cuts the mutant alleles, hence producing two DNA fragments of 225 and 175 base pairs band. Meanwhile, lane 3 exhibits a heterozygous with 400, 225 and 184 base pairs band. It contains both normal and mutant alleles, where it exhibits a total of 3 bands, while one 400 base pairs band from the homozygous normal allele, and 2 bands of 225 and 175 base pairs from the homozygous mutant allele. Do not forget that the total size of digested or cut alleles should be equal to the undigested or uncut allele size. Electrophoresis is usually performed for analytical purposes, where it can be interpreted based on restriction digestion or plasmid DNA forms. Restriction digestion can be defined as the generation of 2 or more DNA fragments by the use of restriction endonuclease enzymes, which cleaves the DNA at the recognition sites. It can be categorized as homozygous or heterozygous. Homozygous shows up as one thick band rather than 2. Each lane should have 2 bands if the sample has heterozygous alleles. Next is based on 4 plasmid DNA forms, which are covalently closed circle monomer, open circular monomer, linear monomer, and OC dimer or concatemer. Firstly, a CCC monomer is an undigested form of plasmid DNA which exhibits supercoiled and intact plasmid which have compact and twisted double-stranded DNA. An example of this form is plasmid DNA isolated directly from bacterial hosts. Next is OC monomer, which is a less compact and supercoiled form of DNA plasmid as compared to the CCC form. It is an open circular form due to a single DNA strand cleavage caused by UV irradiation or nucleases. Linear monomer, on the other hand, is the cleavage of both DNA strands due to restriction endonucleases. Concatemer or OC dimer is an oligomeric form of plasmid which can be due to replication that exists double in size compared to monomers. The second result interpretation is based on the plasmid DNA forms, where when you run a plasmid DNA on the gel, you will probably obtain DNA bands shown in the left figure. Monomer CCC forms move faster than any other forms because they have compact supercoiled DNA structures. Therefore, they will appear further down in the gel due to their intact form, enabling them to migrate faster through the gel matrix pores. The supercoiling process is essential to insert the DNA into the cell, but this form is not accessible for replication procedures. The OC form is an open circular form due to a single DNA strand cleavage, which is caused by topoisomerase, that functions to loosen the tension on it and also provides a free end for the polymerase to work. This form migrates slower and appears very near to the well, as they struggle more to pass through the gel matrix pores than the CCC form. The linear form is obtained when both plasmid DNA strand is cut at one location, making it linear. This form migrates faster than the OC form but slower than the CCC form, hence the appearance of a linear DNA band in the middle of CCC and OC DNA bands. Concatemer form usually moves slower as compared to monomers. This is due to their larger and doubling size, resulting in the appearance of DNA band higher in a gel than monomer. Based on the right figure, it can be concluded that as DNA twist increases, it will result in an increased supercoiling, hence resulting in DNA migrating faster. The figure below is an example of an SDS spatial electrophoresis result where it involves the basis of protein separation according to the mass and charge. For this method, proteins are mixed with SDS detergent, which will unfold the tertiary protein structure into a linear shape and coat them with a negative charge proportional to their molecular masses. Due to their negative charge, protein molecules will migrate toward the positive charge end of the gel and separate according to their molecular masses. The rate of migration of the proteins in the gel is proportional to the logarithm of the molecular weight, where proteins with a lower mass and quantity tend to migrate faster down the gel. Hence, SDS page provides a quick separation of proteins, aiding to the subsequent analysis such as Western blotting.
In conclusion, one of the most commonly used methods in molecular biology is electrophoresis with agarose and polyacrylamide gel. There are straightforward and low-cost approach to separate nucleic acid for quantification and purification depending on site. The agarose gel can be used to resolve large fragments of DNA, while page gel are used to separate a shorter nucleic acid in the range of 1 to 1000 base pair based on concentration used. Both gels consider their own advantage and disadvantage and also the interpretation of results, but also have similarity between them.